This unit is Unit 5, and it is the molecular basis of heredity. It's Competency 5A according to the Department of Education, and we will discuss the central dogma of biology, how traits are passed from one generation to the next. You will be able to draw and label the structures of DNA and RNA, explain the process of protein synthesis, and decipher codon charts to identify amino acids. So first, let's review where all this takes place. It happens in the cell. This is an animal cell. The ribosomes are where the proteins are put together. The mitochondria supplies the energy to the cell that it needs to make the proteins. And the DNA starts out in the nucleus. Erwin Chargaff is credited with discovering the base pairing rule of DNA. So he's the first scientist that we're going to talk about. He discovered that in samples of DNA that he took, that adenine and thymine bases were almost always equal in those samples. In addition, he found that the guanine and cytosine quantities were also equivalent. So he came up with what's called Chargaff's rule, and that's the rule of base pairing. You can see in that picture at the bottom, A equals T, G equals C. Be sure you write that down. We will come back to that a million times in this unit. The next scientist that we need to discuss is Rosalind Franklin. She used X-ray diffraction to try to determine what the structure of DNA. So remember, we said DNA is a huge molecule, and it's got to fit into that tiny space of the nucleus. So she thought if she X-rayed the molecule, that she could discover what it looked like. Watson and Crick are credited with discovering the structure of DNA. So they used Chargaff's rule and Rosalind Franklin's X-ray to discover or to figure out that that molecule was actually a double helix. And that just looks like a twisted ladder. You can't really see it too much in that picture on this slide, but you'll be able to see it when we get working with it in class. So what is DNA? We've been talking about it. DNA holds the genetic code for all living organisms. It's found in every cell in your body. Every cell has an entire copy of the DNA, and that happens, remember, before mitosis. And DNA can also be called the universal genetic code. That is because every living organism on Earth has the same 4 bases: adenine, guanine, cytosine, and thymine. What makes each organism unique is the quantities and how those are arranged in the DNA. So remember, during the S phase of interphase in the cell cycle is when DNA is copied. So before that cell divides, it's got to make an entire copy of all its genetic code and pass that on to the daughter cells that are made. So DNA replication ensures that an exact copy of DNA is made so that none of the genetic information of that organism is lost. You can see the picture on the bottom is showing DNA replication. So in the nucleus, remember DNA never leaves the nucleus, that molecule unzips down the middle, and then there are some enzymes that come in called DNA polymerase. Those go up one side and down the other to form a complementary strand. So remember, we have our base pairing rule, which is Chargaff's rule, A equals T, G equals C. So based on those base pairing rules, the old strand will unzip, that's in blue. The new strand will be formed based on the complementary strands, and that was shown in red on this animation at the bottom. So remember, on the new strand, the 2 new strands are identical to each other, and they complement the existing strand. So the new daughter strands match according to the base pairing rules, A equals T, G equals C. You can see in that picture on the right, each strand has part of the original, the other half is new. So what are the components of the strand? first we have nucleotides. And remember when we discussed organic molecules, we said that nucleotides make up nucleic acids. Those are the monomers of nucleic acids. And every nucleotide has the same basic component. It has a phosphate group, a 5-carbon sugar, and a nitrogen base. And if you remember, ATP is one of the molecules that we discussed that is a nucleotide or a nucleic acid. DNA stands for deoxyribonucleic acid. It is obviously a nucleic acid. Its 5-carbon sugar is deoxyribose. The nitrogen bases are found inside the strand, so those make up the rungs of the ladder. And then the sugar and phosphate combine to form the outside or the backbone of the strand. And you can see that illustrated in the picture at the bottom. The bases are held together by weak hydrogen bonds, and that is for replication purposes and the purposes of transcription. So those bonds are easily broken so that the strand can be separated and the DNA can perform its function. Here we have those 4 nitrogen bases again: adenine, guanine, thymine, and cytosine. As you can tell, that is very important, so I hope you have written that down. So any sequence of bases is possible. And remember, it's those sequences that make us who we are and what we are. So this is DNA replication again. You can see from the picture on the left that strand is unzipping. And for every adenine on the DNA molecule, there has to be an exact thymine. For every cytosine, there has to be 1 guanine. So if the DNA of an organism is 15% adenine, it must also be 15% thymine. Now let's talk about protein synthesis. So the whole purpose of DNA, remember, is to carry an organism's genetic code. But what is that genetic code really for? Well, your DNA codes for the production of proteins. They are very, very important to your health, well-being, and your life. So this little illustration kind of shows the process of protein synthesis. We go from DNA, that is transcribed into messenger RNA, which is sent out to the cytoplasm, that is translated on the ribosome, and then that is made into proteins. So that brings us to the central dogma of biology, and this is the basis of all living organisms on Earth. That is that DNA makes RNA, which makes proteins. And we're going to talk about each step of this process in great detail because there's a lot to go over when we get through it. The way that those proteins are coded and made is what produces your traits. So this is the whole basis of gene expression. So we start with DNA, and then in the end, that produces physical traits that are shown on the inside and the outside. DNA sequences are diverse in each organism. So we have a certain code in humans, but then the bases of that code are also different between every single person on the face of the planet. And we know that because of DNA testing. You know, if you commit a crime, it's very unlikely that a person can share a certain section of your DNA just because of the way that those sequences vary. So the differences in DNA code for different proteins. The variations in the DNA and the proteins that are produced are responsible for variations in physical expression. So you can see all those children at the bottom. They're all children, they're all humans, but such a wide variety in a small sample of just skin color alone shows the diversity of the genetics. Then we go to gene regulation. So those codes are kept in your DNA, but how are they expressed? How are they regulated? Remember, every cell in your body has the same copy of DNA, but every cell in your body doesn't need that entire code. It only needs bits and pieces of it to make the proteins that it needs. So that's where differentiation comes in. So you can see up at the top is a chimpanzee. and then we have a cross section of its thigh. All those different cells that make up that animal's leg are all differentiated. So even though they all contain the entire strands of DNA, all those chromosomes that make up the whole organism, they're only going to use a small percentage of it to make the proteins that they need. So we've been talking about proteins. Why are they so important? Proteins help with your vision. They make up the rods and cones in your eyes. They make up hormones, which can include insulin and pituitary hormones, which code for growth. They are part of your blood. They're the building blocks of life. They form protein channels in your cells. And they are also enzymes that are catalysts. That picture at the bottom is kind of the way that protein is usually depicted. It basically looks like curled up ribbon. So since each cell has a complete copy of the DNA, the entire genetic code is not required to make certain proteins. So different cells make the different proteins and only need sections of the DNA in order to make the proteins that they need. The pieces of the DNA that are used are called codons. And you can see in that picture at the bottom, we have those sections of 3 bases at a time. Those are called the codons. So remember, our DNA contains 4 bases: adenine, cytosine, guanine, and thymine. RNA, however, contains 4 bases also, but instead of thymine, we have uracil. You can read the genetic code by using a codon wheel. That's one of the ways to figure out what that amino acid is. So remember, when we're making proteins, the smallest part of a protein is the amino acid. Those amino acids, the order in which they are placed on that protein strand is going to determine the function of the protein. So let's talk about how we figure out which amino acids are going to be added to the protein. This is one of the ways you can tell, and you will need to know this. You start in the middle. You can see that that wheel is broken down into 4 quadrants. So you start in the middle. We have guanine, adenine, cytosine, and uracil. If we're looking for the amino acid that goes with the codon CAC, starting in the middle, C, then you work out to the second circle in that quadrant, A. Then you go to the third circle in that A section. C is right there, and then that would code for histidine. You may also see a codon table. So this is read in a very similar way, sorta. You start with the first base on the left row, then you move up to the column for the second base on the top, and then for the third base you move over to the right hand row. So if we're looking for AUG, we start on the left, A, then we go up to the top and find the U, then we go to the right and find the G. You follow that over in that row back to the column where the U was, and that codes for methionine, or MET. Coding for proteins begins with the start codon. The start codon is always AUG. So sometimes AUG will be included when you're asked to transcribe or translate. Sometimes it will not. Protein production ends with one of the 3 stop codons. So it's going to keep going down that line of messenger RNA until it gets to the stop codon, which is UAA, UAG, or UGA, and those will usually say stop next to them on the wheel or on the table. Let's look at some of the differences between DNA and RNA. You do need to be able to compare these and contrast them. They're both nucleic acids. They are both made of nucleotides, but they do have some big differences. So DNA, the sugar is deoxyribose. It is 2 strands, contains thymine, and it stays in the nucleus. RNA, on the other hand, its sugar is ribose. It is made of a single strand. It contains uracil, and it leaves the nucleus. The types of RNA, there are 3 kinds. So RNA is what is going to be used for carrying the code to making proteins. It's going to take the message from the DNA and it's going to carry it to other parts of the cell to actually make the protein. So first we start with the messenger RNA. Messenger RNA carries the message. So it takes it down from the DNA, it leaves the nucleus where it goes to the ribosome or the ribosomal RNA. And that's where the proteins are put together. The vehicle for carrying the amino acids is the transfer RNA. So here again, messenger RNA carries information from DNA. It is a single-stranded copy. It's actually a complementary strand of the DNA. The difference is it's going to contain uracil instead of thymine. So wherever that thymine should have been, the uracil will show up. That's a key point in figuring out what type of molecule it is. If it's a single strand and it has U's on that molecule, it is messenger RNA or RNA. So then we have the ribosome where the proteins are put together, and then the transfer RNA is going to carry that amino acid. These are the steps all lined out of protein synthesis. So first we start with transcription. That's going to take down the message. That message leaves the nucleus with the messenger RNA, goes to the ribosomal RNA. Then the transfer RNA comes in with the amino acids. That's going to be the process of translation, which brings in the amino acids to form the polypeptide chain, which makes the protein. So the first step in that process is transcription. Transcription occurs in the nucleus. This is for eukaryotes, obviously, because eukaryotes have a nucleus. The segments of DNA are used as a template to produce the complementary messenger RNA. So you can see that animation at the bottom. The DNA is going to separate. Those complementary bases are going to come in. That messenger RNA is then going to separate from the DNA, leave the nucleus, and go out to the cytoplasm. The messenger RNA is edited so that only the codes for the amino acids are used from the original DNA strand. Remember, we don't have to have the copy or the complementary strand of the entire DNA. We just need bits and pieces to make certain proteins. Messenger RNA, again, is complementary, using those base pairing rules, but does not contain thymine. Enzymes take down the message, substituting uracil for thymine. There's another picture right there. So we have DNA. These are the base pairing rules for DNA. A equals T, G equals C. This is what I want you to write down as soon as you get your test. So DNA codes for RNA, those base pairing rules, A equals T, G equals C, but we don't have T in RNA. We have U. So if you write it down like this, you will know at least that where the T should have been, you should have a U. I find it much easier to remember that way. It gets confusing if you have a long strand to translate or transcript. So here we go again. There's the nucleus, those nuclear pores. Now you finally know what they're for. Now they're coming into play. So the DNA has made that copy of the messenger RNA. It's going to leave the nucleus through that pore and go out to the cytoplasm where it's going to go find the ribosome. Now we can begin the process of translation. So translation occurs at the ribosome. What the ribosome is going to do is it's going to pull in those transfer RNA molecules that are holding those amino acids. So I like to kind of think of it like trains on a track or the track of a tank. So the ribosome is going to kind of roll down that messenger RNA strand, pulling in the transfer RNA based on the 3-letter codons. You can see there at the bottom, we started with AUG, which is the start codon, and it's just going to keep moving down that messenger RNA strand, hooking those amino acids together. So the transfer RNA comes in, brings in all those little amino acids. Those are those dots that you see in that picture on the right, until we have what's called a polypeptide chain. It's going to keep pulling in that transfer RNA and continuing right down that line until we reach a stop codon. That's how the ribosome knows that it's done. After the polypeptide chain forms, it folds in on itself. Remember, the shape of the protein determines its function. That is very, very important. If the protein is not shaped correctly, it's not going to work properly. So that sequence is very important.
What you are about to see is DNA's most extraordinary secret: how a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body. When the RNA copy is complete, it snakes out into the outer part of the cell. Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. and after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.